Hi dear viewers, welcome once again to my YouTube channel and Facebook page. So if you'd like to know how to use FTIR Spectrometer for any kind of perspectives then or any kind of purpose, then this video will be really helpful for you. So in this specific video, I'll try my best to show you how to make a KBR plate or potassium bromide plate for knowing FTIR spectrometer of or FTIR spectroscopy of any compound. So and at the same time, actually how to run FTIR spectrometer by using any computer or laptop, you'll be able to know in the second portion of this specific video. So guys, without any further ado, let's get started. At first, we will clean the each and every components. Those are essential for FTIR spectrometry or this specific tasks. And we are cleaning because if any kind of chemical substance you can say exist here, then it will show extra peak in the you can say IR spectrometer or IR graph. So it will create complexity. So at first we will create you can say that is called KVR or potassium bromide plate for this kind of stuffs. And then we are taking this, it's the you can say in the left hand it's a motor and in the right hand it's the you can say pestle, and we will keep you can say KVR and at the same time some small amount of sample. Then we will mix it, and after mixing properly we will transfer this specific amount of mixture from you can say motor to you can say in the you can say steel pot, where at first we are transferring a small steel component in the you can say larger steel setup, and then we will transfer a small amount of mixture of KVR and sample, and we will use that is called spatula. We will take adequate amount of mixture because we should on our KBR plate more thick or more thin. We have to be careful about it, and we will transfer adequate amount, not very high or not very lower, because it will create complexity in IR graph. So we have to keep in your mind that in this specific segment, because when we will make you can say larger or you can say more thick plate, it will not transfer properly the IR ray, and it will create complexity in the IR graph. And when it will be thick, it will transfer in between the holes of the you can say KBR plate. It will also create complexity. So we have to keep in your mind that, and we will take mixture of KBR and of our sample in adequate amount, not in larger, not in you can say lower. Say yeah, and then we will keep the you can say small steel component in the upper part, and we will press you can say steel rod to transfer into the internal portion, and we will set up you can say pressure machinery. And when we will you can say place in the pressure machinery, we will put in the pressure machine, and when we will properly set up, and we will press, we will use handle to keep the pressure. And you are seeing that it's becoming in the lower portion, and it is pressurized, and you are seeing that it pressure is approximately above 20 atm. When it will approximately 25 to 30 atm, it's enough, and there is no necessity to larger atmospheric pressure to create KBR plate. And that means we have done, and we are successfully completed KBR plate, and we will put this specific machinery on the table, and we are cleaning this specific FTIR cell for and we will press in this portion KBR plate, and then we will use you can say the small ring for properly placing the KBR plate. And we are cleaning because in this specific portion we will keep or place our KBR plate, and if any kind of you can say any kind of component or chemical is exist here, then it will create complexity. And we are pressing in the you can say long glass rod, and we are carefully pressing, and a small steel component has been separated out, and then we are seeing that it's a KBR plate. It is totally uniform, And when we will separated this KBR plate, we have to keep in our mind that carefully we have to separate it out, and otherwise it will, if it is broken down, then it will, we will not able to do FTIR spectrometry. It will create complexity. So slowly and gently we will press here by using this specific glass portion, and we are seeing that it is really uniform, and we can use hand, because it's not, if your hand is you can say clear, then you can use, but it's not you can say suggested, but it is you can say strongly attached. That's why we use hand here, and we will press properly in the center portion of this FTIR cell, and we are pressing spatula to transfer or properly placing. And then after properly placing our KBR plate, we will set up the you can say ring, steel ring. After properly set up you can say ring, we will transfer, we will set up again in the FTIR spectrometer and in the lower portion, and you are seeing that it's a lighting is emitted from left hand side to the right hand side. And in the lower portion, when you will set up, then automatically it is really easier, and you will be able to see it is really easier to set up here. And in this way you'll be able to set up here. And after setting up, we are seeing that the computer screen, and you are seeing there are lots of things. That means sample, measure, view, there are lots of things. We will press measure, and after changing our name, we will press the you can say sample. And when we will press sample and press OK in the pop-up window, then our overall procedure will be ongoing. That means our overall procedure will continue. And then in the lower portion, you are seeing that how many steps are essential, how many scanning is essential for getting proper IR spectrometer. You are saying that scan 14, 15 of 20, that means 20 scanning is essential to get proper or standard IR spectrogram. Means you are saying that it's an IR spectrometer graph, and yeah, it's really awesome here. And we will press say base, we will press any kind, we can choose any kind of specific location, and we can press. Otherwise we can also export, and when you'd like to export, then we will just press save, and in this way we'll be able to know about FTIR graph. So guys, that's all about today. Thank you for